welcome to the third video of your IB Biology Higher Level Topic 8, and in this video 8.3 we're looking at photosynthesis. You'll see as we look at this video we're actually looking at the metabolic process of photosynthesis, and in our next topic, topic 9, we actually look at plants and photosynthesis as such. So we're really looking at photosynthesis here through that lens of the metabolic processes involved. Now, we looked at this in 2.9 and we've been revising this week. So things that you should remember. Okay, photosynthesis is a production of those carbon compounds in cells, and it requires the use of light energy. The main photosynthetic pigment is chlorophyll, and we should know that's found in chloroplasts. That's not even from last year, you should know that from way back in science. Now, last year we looked at photosynthesis through a physics lens, a chemistry lens, and then the biology lens. So as we looked through the physics lens, we were thinking of wavelength. And we said that that visible light from the sun is composed of the spectrum of colours, and that's why we see it as white light. Okay, each colour has its own wavelength, okay, and violet is the shortest wavelength, whereas red is the longest wavelength. And what we said was not all of the light is used in that photosynthetic pigments. Mostly green is reflected, and we said the red and the blue are absorbed more than other wavelengths. And then we looked at the photosynthetic spectrum to go further into that. So the first stage of photosynthesis is where that absorption of light occurs, and that involves chemical substances that we call pigment. And one of those pigments that you know or have heard a lot about is chlorophyll. Okay, now there are many different pigments in the organisms that absorb light, but yeah, as I say, chlorophyll is that one that you know lots and lots about, found inside the chloroplasts, and we will be looking further into the chloroplasts either later in this unit or in topic 9. Okay, so you will remember the absorption spectra that you saw back in 2.9, showing those different wavelengths and those higher absorption rates. Also, in this graph here, you can see the difference between that chlorophyll A and chlorophyll B, just with slightly different absorption rates. Another graph that we looked at is what we call the photosynthetic action spectrum, okay? And this was where we looked at the rate of photosynthesis at different wavelengths. So comparing those two graphs, on the previous graph that I just had up, you had the wavelength across the bottom and that rate of photosynthesis. Here you've got the percentage absorbed against the wavelength. Okay, so relative photosynthetic rate there on your vertical axis. Comparing the two there, one step ahead of myself. All right, so photosynthesis and the chemistry. Okay, so what we're looking at is the production of these carbon compounds in cells using light energy, and we've got our chemical formula there. Sorry, our chemical equation. So we've got our reactants, which are carbon dioxide and water, our products, that's sugar and oxygen, and this chemical reaction can only occur with the presence of light energy, but also the presence of chlorophyll. Okay, so we said that we had two different stages, our light dependent and light independent stage of photosynthesis. This is all from 2.9, so still a revision. So in our light dependent stage, light was absorbed by chlorophyll. The energy from the light was used to split the water molecule, and we called that photolysis. So we got our hydrogen ion and an oxygen molecule, remembering that has the two oxygens. The oxygen was then released as a waste product, or is released as a waste product. So all the oxygen generated by photosynthesis actually comes from that photolysis of water. the hydrogen ions, they now get carried on to that next stage of photosynthesis. So this is our light-independent stage, where the hydrogen ions have arrived from step 1. Carbon dioxide is now used. The carbon, the oxygen, and those hydrogen ions are combined to make our glucose. The production of this complex molecule requires energy to create those bonds. Okay, and we're going to look further into that now. Okay, so our new stuff, some nice stomata there in the background. Alright, so we're going to look further into the chloroplast. So there you've got a diagram of the chloroplast. And here we've got a diagram of the chloroplast. And this is one of the things that you need to be able to do, is draw a diagram of a chloroplast. So, thinking of our chloroplasts, let's have a look at their structure and their function. So these are variable in structure, but they do have many similarities between one chloroplast and the next. They all have a chloroplast envelope, and this consists of a double membrane. They have thylakoid stacks, which are called grana. This is an intense green internal membrane, okay, and you can see that in the diagram there. They contain small fluid-filled spaces. Okay, so you can see those in the diagram there, and you've got that stroma, which is that aqueous fluid there. Okay, so our stroma, which we're now going to talk about, this is the colourless fluid around the thylakoids containing a range of enzymes. Okay, it's got a real soup of enzymes in it. It might also contain some starch grains and also some lipid droplets. Okay, so that's having a look at your chloroplasts. Now let's start having a look in a bit more depth at this process of photosynthesis, this metabolic process, and we're going to start looking in more depth at that light-dependent stage. So this occurs in these thylakoids, so the stacks, the grana, okay, these stacks of double-membraned organelle as such. Okay, so looking in a bit more depth here, we're looking at the actual membrane. Okay, so you've got the chloroplast stroma. This is outside of the thylakoid, and then you've got the thylakoid membrane, and then inside. We're going to look at this all in a bit more depth, but you can see that there is quite a complex process occurring here. Okay, these metabolic processes are quite complex. Okay, so in this light-dependent reaction, the purpose is to produce NADPH for the light-independent stage of photosynthesis. So this occurs in the chloroplasts of the cells. It requires chlorophyll to occur. Chlorophyll is located in the thylakoids and arranged into two specific structures, which are called photosystems. We've got photosystem 1. Okay, you'll see it's actually second in the systems, but photosystem 1 and photosystem 2. So photosystem 2. Okay, so we're going to look at photosystem 2 first, because that's actually the first system that occurs. Alrighty. In photosystem 2, this is what we call photoactivation. Okay, so photosynthesis begins with the photoactivation of the photosystem 2. Light energy from the sun is absorbed by the chlorophyll molecules in PS2, which gives electrons in PS2 a large amount of energy. So it's all about that light energy exciting the electrons. These electrons are said to be excited and are promoted to a higher energy level because they've got more energy. When they are at this higher energy level, they are captured by the primary acceptor of the reaction center. Okay, so you've got the reaction center here. You've got these excited electrons, and they're captured into that reaction center. Once the electrons have been excited, they pass along the electron transport chain within the thylakoid membrane. They pass between the carrier molecule plastoquinone, which is known as PQ, to a cytochrome complex. And then they go to what we call plastocyanin, PC, and finally to photosystem 1. As they move along, the electrons lose energy, okay? This energy is used to pump protons, so our hydrogen ions, from the plant stroma into the thylakoid lumen, that thylakoid space. Okay, so we've got our electrons becoming excited, they go into the system there, and then they move along the chain over to the photosystem 1, and that's our electron transport chain there. After a while, there is an accumulation of protons in the thylakoid space. A concentration gradient for protons is therefore established. Protons then diffuse across the thylakoid membrane back into the stroma via chemiosmosis. So back to the stroma, back across by chemiosmosis. As this happens, ADP is phosphorylated to form ATP. It is this reaction that is known as photophosphorylation. So we've heard about photophosphorylation before, and this is what it's all about. Okay, so photolysis, where does that come into things? Well, water is split by an enzyme to produce electrons, hydrogen ions, and an oxygen atom. So we know that we talked about this photolysis. This process uses the energy from light and is called photolysis. The electrons are supplied to the photosystem 2 to replenish those lost from photosystem 1. So you see it's actually a continuous cycle. We say photosystem 2, photosystem 1, but it's actually this continuous metabolic cycle. All right, so let's have a bit more look at photosystem 1. So the excited electrons captured by the plastoquinone are passed on to other carriers that ultimately end up at photosystem 1. A photon of light is absorbed in the chlorophyll in photosystem 1. This results in an electron with a higher energy state, which is transferred to the primary electron acceptor in the reaction center. So remember, the reaction centers are in the center here. Again, this is called photoactivation. The electrons from the higher energy state are passed down the electron transport chain to the carrier ferredoxin. The ferredoxin is used to reduce the NADP to NADPH, which is then used in the Calvin cycle. Okay, so we're seeing that thylakoid again. You've got lots and lots of things going on here and structures that you've just heard about. You've got your photosystem 2, your photosystem 1, ferredoxin, you've got your NADP reductase, which is the enzyme that reduces the NADP. You've got your stroma, the lumen of the center of the thylakoid. You've got your plastoquinone, you've got your plastocyanin. Yes. Okay, so an overview of the photosystems, photosystem 1, photosystem 2, our overview. Electrons excited in photosystem 2 are passed along the chain of carriers to our plastocyanine, which transfers them to photosystem 1. The electrons are re-excited with some light energy and are eventually used to reduce NADP. Alrighty, so cyclic photophosphorylation. Sometimes the supply of NADP runs out. When this happens, electrons return to the electron transport chain that links the two photosystems, and rather than being passed to NADP, as the flow back along the electron transport chain, they pump electrons into the thylakoid space. This allows ATP production via chemiosmosis. Alrighty, so now we move on to that light independent stage in a bit more depth than we did in 2.9. So, the light independent stage occurs in the stroma, so not in the thylakoids now, outside in the stroma. Right, so we know this is the stroma here, this is the thylakoid membrane, and then you've got the thylakoid lumen to the center of the thylakoid there. So our light independent stage is occurring out here. Let's have a look at this in a bit more depth then. After the light dependent reaction has occurred, there is a light independent reaction. We know that. This involves the product of the light dependent reaction, NADPH. The initial substrate of the light independent reaction is the 5-carbon compound ribulose biphosphate. We call that RuBP. RuBP is also the product of the light independent reaction, which is why this series of reactions is a cycle, okay, and we call it the Calvin cycle. So the initial substrate is the same as the product, so it's a cycle. And there it is in its glory, the Calvin cycle. In the first step of the cycle, RuBP combines with carbon dioxide, which has entered into the stroma of the chloroplast by diffusion through the stomata of the leaf. The combination of carbon dioxide and RuBP is a carboxylation reaction, which is catalyzed by the enzyme RuBP, carboxylase, or we call it Rubisco, and is commonly called carbon fixation. The product of this carboxylation is a 6-carbon compound, you do not need to know the name, which instantly splits into 3 carbon compounds called glycerate 3-phosphate, G3P. Ribulose biphosphate is then regenerated so that the cycle can restart again. In order to regenerate the RuBP, several triose phosphates are linked together. For every 6 triose phosphate that are generated, 5 are used to regenerate the RuBP. So we've got your triophosphates over here. RuBP is a 5-carbon compound, so 5 triose phosphate molecules are required to make 3 RuBPs. You can think about this mathematically. 5 TP is 15 carbon, so 15 times 3 is, 5 times 3 is 15, and 15 carbons is 3 RuBPs. 15 divided by 5 is 3. And finally, It takes 6 cycles to produce 1 glucose molecule. Alrighty, so a summary of the Calvin cycle. RuBP is combined with CO2 via carbon fixation catalyzed by rubisco to form a 6-carbon compound that immediately splits into 2. The 6-carbon compound splits into 2 3-carbon molecules called glycerate 3-phosphate. Glycerate 3-phosphate is then reduced into triose phosphate TP. This reaction requires ATP and hydrogen ions, which come from NADPH as it is oxidized back to NADP. One of 6 triose phosphates from each cycle combine with another TP to form a molecule of glucose. 5 of 6 triose phosphates combine to form 3 molecules of RuBP or RuBP. Okay, so this cyclic reaction was investigated by a scientist who we now know as Calvin, okay, and his team established the details of carbon fixation. They used advanced lab equipment, such using some really new technologies in the day, radioactive tracers and audio radiography. We've heard of audio radiography before. The team developed the lollipop apparatus that you see there in the picture. The vessel is designed to mimic the shape of a leaf, and it's broad and thin, so this is the lollipop here. Okay, so Calvin's experiment then, let's have a look in a bit more depth of it. Chlorella algae was placed in the lollipops. You've got the lollipop vessel there. The algae was exposed to carbon-14, which is a radioactive isotope, and light. Samples of the chlorella were then released from the apparatus in short intervals. The released sample was placed into boiling methanol. This denatured the enzymes and halted photosynthesis. The compounds in the algae were then separated using 2-way chromatography paper. The radioactive compounds were then identified using autoradiography. Calvin realized that the algae at different time Calvin released the algae at different time intervals. This meant he obtained products at different times. This allowed him to sequence the steps of the Calvin cycle. And there we are. That is this section on 8.3 completed. We will go into more depth on some of these areas as we study topic 6.